Remember that a sarcomere is a contracting unit of a muscle fiber. The muscle fiber is composed of myofibrils. And so a myofibril can have 10,000, up to 10,000 sarcomeres kind of lined up end to end, all the way down the length of it. A sarcomere is defined as the region between two Z-lines. So Z-lines mark the boundary at the end of the sarcomere, on the left and the right end of it. The little green protein that was kind of wavy in the image is called titin. Titin connects the Z-lines to the thick filament or the myosin. The M line is at the center of the sarcomere, and it also forms the central portion of the thick filaments or the myosin filaments. Then we have bands. The A band, remember the word dark has an A in it. So the A band is the darker region of the striations because it is composed of the length of the thick filaments or the myosin. The I band is the lighter region, and remember the word light has an I in it. And it's lighter because it contains the thin filaments or the actin. So it's going to include the actin filaments on either side of every Z line. And the H band is going to be the region that only contains thick filaments. So the H band is within the A band, but it's the portion that's only myosin. There's no overlap of actin. And the size of the the size of the H band will change when it will get smaller when the sarcomere contracts. Now, the thin filament itself consists of actin. At rest, the active sites on actin, there are active sites that myosin is going to bind to. The active sites are covered by strands of tropomyosin. And then the tropomyosin strands are held in position by troponin. So it's the troponin that has to kind of move out of the way so the tropomyosin will uncover those active sites so that myosin will be able to bind to actin and a contraction can occur. The thick filaments are called myosin. They have a tail and they have heads, and the heads interact or bind with the active sites on actin during a sarcomere contraction. The way the active sites are unlocked, and what we mean by unlocked, remember, is there's a molecule called troponin that is holding tropomyosin over those active sites, so they can't, the myosin heads cannot bind to the actin. So calcium, this is where calcium comes in. Calcium ions unlock those active sites by binding to troponin, and that allows the tropomyosin to move out of the way of the active sites. Now, this is all the individual portions of the sarcomere organized for you. And so the only thing I'm going to show you is just, I want you to focus on the strand of actin, which is really two strands, like two strands of beads that are twisted. All right, this rope-like greenish blue rope-like structure is the tropomyosin, and the troponin is this brown molecule that's holding it in place over the active sites. Those active sites on actin are yellow. They're yellow portions of the red beads. So the tropomyosin covers the active sites. It is held in place by troponin, and what moves the tropomyosin off of the active sites is calcium binds to troponin. Troponin causes the tropomyosin to move out of the way. And now the active sites are exposed, and the myosin head can bind to them and cause the contraction of the muscle fiber. So, the explanation for sarcomere contraction is called the sliding filament theory. The sliding filament theory. And it's based on observed changes in the sarcomere. What we observe happening during the contraction is that the I bands get smaller, the light bands get smaller, the Z lines move closer together, The H bands also get smaller. They decrease. The zones of overlap get larger. So in other words, the actin overlaps even more with the myosin. But the length of the A band, which is the length of the myosin filaments, remains the same. So it seems like the actin is sliding over the myosin. So here's the conclusion, is that thin filaments, or actin, are sliding toward the center of the sarcomere, and thick filaments are just remaining in place. And this is what a muscle fiber looks like relaxed before the sarcomere contracts, and then what the sarcomere looks like after a contraction. So you can see before the contraction, the length of the A band is the length of the myosin filament. You can see the length of the I band between the Z line and the start of the A band. You see the length of the H band, which is just the portion of myosin that has no overlap with actin. Okay? After the contraction, that H band is smaller. The I band is smaller. So it just appears that the actin filaments, these thin filaments, are moving toward the end line, toward the center of the sarcomere, and the thick filaments are just staying right where they are. And that titin is kind of like a spring, you know, it's just bunching up a little bit more when the actin filaments move toward the middle of the sarcomere. So that leads us to the mechanism or the steps of the sliding filament theory. In other words, what actually is causing the sliding filament theory or causing the contraction to happen? All right, well, we know myosin heads. The myosin is the thick filament. So the heads of the thick filaments bind to active sites on actin, which are the thin filaments. Once that binding occurs, we call that part where the myosin heads are bound to the active sites, we call that a cross bridge. So we call that cross bridge formation when myosin actually binds to actin. Then those myosin heads can pivot, and they can pivot toward the center of the sarcomere, kind of pulling actin closer to the center of the sarcomere. And that is the, in some of the video tutorials that you have, like the interactive physiology, they'll call that the power stroke. Once that happens, that is the contraction. Now the cross bridge is going to detach, and that is where ATP comes in. ATP allows the cross bridges to detach. It allows the myosin heads to be removed from the actin and return to their original position. Checkpoint question. Where would you expect the greatest concentration of calcium ions to be in a resting skeletal muscle fiber? And I would say that will be the terminal cisternae. Neuromuscular junction. All right, so we already discussed the steps of the muscle contraction itself, but what leads to the muscle contracting? What leads to the calcium being released so that it can bind to the troponin and uncover, the tropomyosin can uncover the active sites? What causes that to start? Because the muscle cell is at rest. So what's going to lead to that muscle cell, to all of those steps of the muscle contraction beginning? And that's going to have to do with a motor neuron. That's a nerve cell. Remember, a neuron is a nerve cell. So that's going to be the axon of a nerve cell called a motor neuron, and its interaction with the skeletal muscle fiber or the muscle cell. Okay, so what really happens involves the axon terminal of the motor neuron. So remember the end of the axon, which is called the axon terminal, it's kind of an expanded end. It's just kind of swollen, and it contains these vesicles that contain chemicals called neurotransmitters. So the neuromuscular junction consists of an axon terminal of the motor neuron, and then what we call the motor end plate of the muscle fiber. And that's just the portion of the muscle fiber we call that, remember, the sarcolemma. That's the portion that is interacting with the axon terminal of the motor neuron. So we just call it the motor end plate. So the axon terminal of the neuron contains vesicles filled with a neurotransmitter called acetylcholine, or ACH. And that is going to be the stimulus for the muscle fiber contraction. The motor end plate is the portion of the muscle fiber sarcolemma. So this is what happens. The events at the neuromuscular junction that are going to lead to a muscle contraction start with acetylcholine being released from the axon terminal, diffusing across the gap between the neuron and the muscle. That's called the synaptic cleft, because what's happening here is called a synapse. So the synaptic cleft is just a gap between the nerve and the muscle, the axon terminal and the motor end plate. So acetylcholine is released into that synaptic cleft, and it binds to a receptor on the motor end plate. Now, the enzyme acetylcholinesterase, ACHE, is going to break down acetylcholine so it doesn't keep on stimulating the muscle to contract. So it's going to break it down so that it goes back into the motor neuron and doesn't continue to stimulate the muscle, okay? So that's why that's in here. The acetylcholinesterase is the name of the enzyme that breaks down acetylcholine. So if it doesn't break it down, it's just that acetylcholine is just gonna keep binding to receptors, and the muscle won't stop contracting. And that can be very, very bad too. All right, once the acetylcholine binds to the receptor on the motor end plate, remember, that's the sarcolemma of the muscle fiber. Then an action potential or electrical impulse is generated in the sarcolemma, and it's going to travel down those T-tubules to the interior of the muscle fiber, and then the whole muscle fiber is going to contract. So I'm going to have to blow this up. These are the events at the neuromuscular junction, just in a little more detail. Okay, so what you have going on here is this is the axon this is a motor neuron. Motor neurons are multipolar neurons. They have a bunch of dendrites extending out from the cell body, and then an axon, a single axon. And the axon has all these different branches at the end of it, but the portion that actually interacts with the muscle fiber is called the axon terminal, and the muscle fiber portion is called the motor end plate. So let's blow that up. Here's the axon terminal, and here's the motor end plate. And here you see the vesicles that contain acetylcholine. The acetylcholine being released by exocytosis into the synaptic cleft between the axon terminal and the motor end plate. And the acetylcholine is binding to receptors on the sarcolemma, the motor end plate itself. And then once that happens, it's going to generate an action potential. Let's look at the next page. And that action potential, okay, so let's look at step 3. When the action potential reaches the axon terminal, the action potential in the neuron reaches the axon terminal, the acetylcholine is released into the synaptic cleft. That's step 3 here. And then exocytosis occurs. The vesicles release the neurotransmitter acetylcholine into the synaptic cleft. Step 4, the acetylcholine molecules bind to the receptors on the surface of the motor end plate here. And that leads to the membrane becoming highly permeable to sodium. So sodium rushes in, sodium ions rush in, and in step 5, this results, because of the rushing in of the sodium ions, this changes the voltage or the potential in the sarcolemma of the muscle fiber from -70, and it starts going more positive because sodium ions are positively charged. So once sodium comes in, it changes that voltage from -70 to a more positive number, like -55. And that is going to generate an action potential. That's like the threshold level that it has to be to generate an action potential. And then more sodium is going to rush in. And that's going to lead to an action potential, and it's going to travel all the way down the length of that muscle fiber. So that's the nerve impulse portion. And then what happens after that is the sliding filament theory, the actual muscle contraction itself. And the contraction cycle is the link between that action potential generation and the start of the muscle contraction. So what the motor neuron does is generates an action potential in the muscle fiber. And after the action potential, that will stimulate those steps of the muscle contraction, which involve calcium binding to troponin and then tropomyosin repositioning and moving off of the active sites so that myosin can bind to actin. So the action potential travels over the sarcolemma, down the T-tubules, and this causes the release of calcium from the terminal cisternae. Calcium binds to troponin, and the contraction cycle starts. And so the steps here is just going to say calcium binds to troponin, exposing the active sites on actin. The myosin heads come up and bind to the active sites. That's cross-bridge formation. And then the myosin heads pivot. You see how they're like this, and then they pivot. That pulls the actin closer to the center of the sarcomere. And that's the contraction itself. So what releases those heads is ATP. ATP comes in, and that causes the breakdown of ATP, causes the release of those myosin heads from the active sites.